So one of the main things that we've been working on is to develop and apply new genome editing technologies to understand genetic contributions of different kinds of disease. And so this will be the main topic for today's discussion. So one of the major advances in the field of biological research in the past decade is the rapid development of genome sequencing technology. So we all have a genome, and the genome is like a blueprint for how our body is built and how we are able to live a life. And so from these sequencing studies, we're starting to elucidate different genetic differences that may or may not contribute to specific kinds of disease. And so this is a plot released by the National Institutes of Health. And you can see over the past almost 10 years, the number of genetic variations that have been identified has really grown exponentially. But one thing to remember from this plot is that these differences are not causal. We don't know if the presence of the mutation necessarily lead to the disease state. All we know is that they are linked. They are found in some of the people who have these kinds of conditions. And so what we need is to be able to understand and also identify which one of these mutations actually cause disease. And so fortunately, the work of many, many laboratories have started to shed light on this. And so over the past several decades, we have started to elucidate single gene mutations that contribute to many different kinds of disease. But this is really just the surface of all of the different kinds of genetic mutations that can underlie a variety of different pathological conditions. And so what we need to do is to increase our understanding of genetic mutations. And through understanding genetic mutations, then we can develop new kinds of therapeutic treatments. And so what we need to do to understand the genetics is to develop new tools. And the type of tools that we have been developing is by looking into nature and then getting inspirations from the things that exist out there that we can then borrow and then use it to do very useful things to understand biology. And so if you look at this picture of the forest out there, there are thousands or tens of thousands of different kinds of large organisms, microorganisms, and they, in order to survive in nature, they evolved very different kinds of mechanisms that we don't possess. But if we understand what they are and then harness them, then we can use it to understand how our own body functions. And so errors in the genome can lead to many different kinds of dysfunctions. And so what we need to do is to identify the functions of the genomic sequences and the functions of variations. And also, if we know what these variations are, then we can develop new treatments. And maybe one of the treatments is, if we know one mutation causes cystic fibrosis, why don't we just go in and then replace it with the correct sequence so that we can rescue the disease. But many of the existing strategies don't work as well because they don't actually go into the genome and fix the mutation the way that we need it to be fixed. And so this is one example. So if you take this poem from Robert Frost, and there's a typo there. It's supposed to be yellow, but it's actually blue. So in order to fix it, there are many different ways. The existing ways, one of them is simply to get rid of it. So now if we get rid of that word, the poem doesn't make sense anymore. So it's not the right way to fix it. Another way that exists today is to add in the correct word, but it doesn't get rid of the existing word. So you have redundancy, but that also changes the meaning of what you're trying to do. So what we really need to do is to go into the genome, find that mutation, and specifically replace it with the correct version so that we go back to what the intended meaning of the sentence is supposed to be. But in the cell, the genome is hard to do it. We don't have a search and replace function. And so what we need is to develop a new way to actually search for that error and then replace it. And so it turns out that nature has evolved these kinds of tools, the search and replace function. And that is called a DNA binding protein. And so this is a small protein that can go into the genome and then based on the sequence, latch onto the exact location that you want to latch onto. And so with this DNA binding protein, then you can build scissors that can literally cut and paste within the genome. So to overcome some of these challenges, we and others have started to develop a different technology called CRISPR. And CRISPR was originally found in bacteria that were used to make yogurt. And it turned out that this mechanism called CRISPR is what some of the yogurt bacteria are able to use to defend themselves against viral infections. You don't want viruses to infect the yogurt because it'll spoil the yogurt and it wouldn't taste very good. So it turns out that in nature, if this is a bacterial cell, many different viruses can invade this bacterial cell. And so what happens is the bacterial cell wants to develop an immune system to be able to fight against these new virus infections. And the way it does this is using a system called CRISPR, where small pieces of the genetic information from these viruses are recorded into the genome of the bacteria. Using this methodology, we have been developing ways to see, can we turn this into a therapeutic? And turns out that we can use different kinds of methods to deliver this into tissue within the animal body. And so using viruses, we can package these CRISPR-Cas9 reagents and then deliver it into, for example, the liver of a mouse. And then within the liver, we can target genes that can treat hypercholesterolemia or target genes that may be involved in hemophilia. And so these are some results that show that within a short period of time, within 3 to 7 weeks, we can achieve pretty efficient modification of the genome within the liver of the mouse. And so this is really just the beginning, but the potential is really enormous, because one of the very promising examples out there is that people have been able to use genome editing to treat HIV infection. And the idea is that based on the story of this Berlin patient, Timothy Brown, the HIV virus usually infects the cell through 2 different proteins. These are 2 different receptors on the immune cell. And turns out that one of the receptors, CCR5, is not so important for a person to live a very healthy life, because there are individuals who are naturally devoid of the CCR5 protein. So what that means is that we can get rid of the CCR5 using genome editing, and then the virus wouldn't be able to infect the patient. And so there are currently clinical trials using older versions of technology to achieve this kind of editing process to treat HIV. But we can certainly use CRISPR to treat not only this, but many other kinds of genetic diseases where modification at the genomic level will be efficacious. I'm really grateful for the Waterman Award, which is going to enable us to do additional levels of development, not only on the CRISPR technology, but also look into new areas that are going to be too risky to obtain funding from traditional mechanisms. Thank you very much.